Metabolism refers to the chemical processes and reactions that help living organisms grow and reproduce. This includes the ability to break down nutrients, like in a cookie, as well as the ability to rebuild different nutrients and structural components. We have seen how energy coupling allows cells to transfer energy from high energy bonds in food into ATP, which can then donate this energy to anabolic reactions. We have looked at ATP. We have looked at the basic patterns of energy that life uses. Now, it's time to dive into exactly how that energy is transferred from the food that we eat to ATP. This is the study of three catabolic pathways: glycolysis, the citric acid cycle, and the electron transport chain. We will look at the reactions and catalyzing enzymes within each process in the hopes of really understanding how energy flows from a cookie to ATP. This is the study of three catabolic pathways: glycolysis, the citric acid cycle, and the electron transport chain. We will look at the reactions and catalyzing enzymes within each process in the hopes of really understanding how energy flows from a cookie to ATP. Here's an overview of the three cycles that we will look at in this chapter. You have learned a lot of information that will help you understand what is going on in these reactions. First, glucose is broken down into two pyruvate molecules. This step provides the cell with a net gain of 2 ATP. However, this is not nearly enough ATP for multicellular organisms like ourselves. We need more. Lots more. So the two pyruvate molecules are converted into acetyl coenzyme A, or just acetyl-CoA. Coenzyme A is a carrier molecule. The important part is the acetyl group. This leads us into the citric acid cycle, or the TCA cycle, as it is lovingly referred to. It is called this because it involves citric acid, or citrate. Acids that have lost their proton are often named with the ATE at the end. So citric acid becomes citrate, and glutamic acid becomes glutamate. It's just a fancy way to say that it has lost its proton and become charged. Each acetyl-CoA goes through the citric acid cycle, which produces 2 CO2 and 1 ATP, as we would expect. But it also produces a new molecule which we haven't seen before, NADH, which we will look at the structure of this molecule more later. For now, we just know that it is an energy carrier. The energy from NADH, in the form of electrons, is transferred to these four enzymes and ultimately produces a gradient of protons across the mitochondrial membrane. We've seen what happens when there is a higher concentration of molecules on one side of a membrane than the other. If we open a channel, the molecules move from higher concentration to lower concentration. We know this is diffusion. Here, the difference is so great that lots of protons want to get across the membrane to the lower concentration area. This creates what is called a proton motive force. These protons need a way across the membrane. Fortunately for us, a final, massive enzyme complex, the ATP synthase, gives them a way across. The proton motive force is used by this pump to turn the massive protein complex at its base. This is like water falling over a wheel. The force of the water moving across the wheel pushes it around and around. The turns of this massive protein complex convert ADP and a phosphate group into ATP. This is the magic of cell biology. Glucose has been broken down to provide the proton motive force to turn the wheel of ATP synthase and provide the cell with the energy for life. So, there are two new important molecules that we need to learn about before we can look at the details of these reactions. They are NADH and FADH2. We can talk about them together because they basically do the same thing. They start out in what we would call their oxidized form. They will participate in what are called oxidation-reduction reactions. Oxidation-reduction are the processes that describe electron transfer from one molecule to another. In Chapter 2, we talked about some atoms can steal the electron from another. In this case, electrons are passed around between molecules as a way to store energy. NAD+, when combined with 2 electrons and 1 proton, make NADH. So when we see the formation of NADH, we know that electrons have been transferred for later use. Gaining electrons to become NADH is called reduction. One way to remember this is that electrons are negatively charged, so we are reducing the overall positivity of the molecule. The opposite of that is to be oxidized. Oxidation means that a molecule lost electron. Oxidation loses electrons, and reduction gains electrons. So we know about ATP and NADH, and we are ready to dive into the first of our three pathways, glycolysis. We're going to go through this, reaction by reaction, using this format. You need to know the type of enzyme listed here at the top, the name of the products, and whatever is lost or gained by the reaction. We start with glucose. Glucose is converted to glucose 6-phosphate by a kinase enzyme. Kinases add phosphate groups stolen from ATP. Once phosphorylated, glucose can't readily leave the cell. This reaction is exergonic, and the enzyme is allosterically regulated. This reaction is not reversible, so we have a single directional arrow. Watch the arrow for reactions that are reversible. The second reaction is a rearrangement of glucose 6-phosphate into fructose 6-phosphate. The enzyme that catalyzes this reaction is an isomerase, and it is reversible. Notice that nothing has been added or removed, just rearranged. We see that the ring oxygen is now bonded to carbon 2 instead of carbon 1. This is an isomer, hence isomerase. The third reaction is another kinase reaction. ATP donates a phosphate group again, and we have fructose 1,6-bisphosphate. Bis just means 2. So far, we have put 2 molecules of ATP into this process. So we haven't seen any benefit yet. The next reaction is catalyzed by an aldolase. In this step, fructose 1,6-bisphosphate is basically ripped in half. We can see that the 2 product molecules both contain 3 carbons, and that they are isomers of each other. In fact, another isomerase can switch them around. This is what happens, so we end up with 2 copies of glyceraldehyde phosphate. Every reaction from here on out is actually happening twice, because we now have 2 molecules to proceed with this. This 2 at the bottom will help remind you of that. Next, glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. This is an interesting reaction. We see here that NAD+ is being reduced to NADH, which means that glyceraldehyde 3-phosphate is being oxidized at the same time that a free-floating phosphate group is attached to it. Because this reaction happens 2 times, we end up with 2 NADH molecules carrying electrons that we will use up later on. We've reached the stage where we can start getting back some of the ATP we spent in the beginning. Remember that glycolysis has used 2 ATP so far. However, in this step, we remove one of the phosphates from 1,3-bisphosphoglycerate and use that to turn an ADP into an ATP. Alright, we have broken even. This is a kinase reaction, and it leaves us with 3-phosphoglycerate. The next reaction is catalyzed by a mutase, which is really just an isomerase that moves phosphate groups. Here, we have another isomer, where 3-phosphoglycerate has become 2-phosphoglycerate. In the 8th reaction, we lose a water from 2-phosphoglycerate and form phosphoenol pyruvate. This is only one step away from the final reaction in glycolysis, and so far, we have no more ATP than we started with. Let's see what happens next. We've made it to the final reaction. In this step, phosphoenol pyruvate is converted to pyruvate by a kinase reaction. Pow! The last phosphate group is pulled off and added to ADP again for the production of ATP. We now have 2 more ATP than we started with. But we also have 2 pyruvate molecules. Ultimately, we want to convert our starting glucose to CO2 to get as much ATP out of this as we can. So these 2 pyruvate molecules are going to set out on a quest to generate even more ATP until nothing is left but CO2. The quest has led them here, to the very walls of the mitochondria. By passing through into the heart of this energy production organelle, the pyruvate will be changed into acetyl-CoA and give off our first CO2 molecule. Pyruvate crosses both membranes of the mitochondria and ends up inside, where it is converted to acetyl-CoA. Remember that the coenzyme A is just a carrier molecule for the acetyl group. In order to give off a CO2 molecule, pyruvate was oxidized, causing the reduction of NAD+ to NADH. The acetyl-CoA created in this reaction will be fed into the citric acid cycle. Remember that this happens 2 times for each glucose molecule. The citric acid cycle overview is given here. We see that as the molecules go around the cycle, they give off 4 CO2, 6 NADH, and 2 FADH2 molecules. We can also see that the last molecule in the cycle, oxaloacetate, is used to start the next round. It will be important for you to know which molecules are created at each step and what products are given off. You do not need to know the names of all the enzymes in this cycle, just the products and reactants. However, it is important to know that many of the enzymes in this cycle are allosterically inhibited by the products, which means it's a negative feedback loop that keeps the system regulated. So be aware of which steps are important for regulation. The first step in the TCA cycle is when the acetyl group from acetyl-CoA is incorporated into oxaloacetate, forming citrate. Citrate, the ionized form of citric acid, is where this cycle gets its name. Then, citrate is isomerized to isocitrate, which leads us to one of the most important steps, the oxidation of isocitrate into alpha-ketoglutarate. This step accompanies a loss of CO2, a proton, and the reduction of NAD+ to NADH. The enzyme in this step, isocitrate dehydrogenase, is a primary point of regulation. It is allosterically enhanced by ADP, which makes sense since its job is to make ATP. However, NADH is a competitive inhibitor of this enzyme and slows the cycle down. NADH is creating a negative feedback loop and ensuring the reactions don't overdo it. The enzyme in the next step is also an important regulatory point in the cycle, and is allosterically and competitively inhibited by NADH and ATP. In this step, alpha-ketoglutarate is oxidized into succinyl-CoA by the addition of a CoA carrier molecule. This is another step where we get CO2 and NADH. Next, we have the conversion of succinyl-CoA into succinate. This reaction is coupled to the conversion of a GDP to GTP. GTP, as we've seen before, is similar to ATP. Succinate is oxidized into fumarate, and FAD is reduced to FADH2. Again, it is important to know that oxidation and reduction reactions are coupled. So as one molecule is oxidized, that is, it loses electrons, another molecule picks up those electrons and is reduced. In the next step, water is added to fumarate to form malate. Malate is an important molecule in other metabolic processes like gluconeogenesis that we will look at later. In the final step, malate is oxidized to oxaloacetate, and another NADH+ is reduced to NADH. This oxaloacetate can then be reinserted into the cycle by addition of another acetyl-CoA. In fact, this whole process happens twice because one glucose produces 2 pyruvates, which then each produce an acetyl-CoA. So out of the TCA cycle, one glucose produces 2 GTP, 6 NADH, 2 FADH2, and 4 CO2. If we add this to glycolysis, we have an additional 2 ATP and 2 CO2. This means the cell has completely converted all 6 carbons from glucose into CO2. We have only directly generated 4 ATP molecules so far. This is not nearly enough. We need to look at what fate awaits the other products from these 2 pathways, NADH and FADH2. They are headed to the inner mitochondrial membrane, where they will hop aboard the electron transport chain, a pathway to the home of the ATP synthase, and a whole lot more energy production. There are 4 proteins involved in the electron transport chain. These proteins are referred to as complexes 1, 2, 3, and 4. And they are active transporters that pump protons from the inner mitochondrial matrix to the inner membrane space. They are assisted by 2 carrier proteins, coenzyme Q and cytochrome C, which both act to carry electrons between complexes. Here we are at complex 1. The first molecules to arrive are NADH. NADH is then oxidized to NAD+ by complex 1, which is reduced. This means that the 2 electrons from NADH flow to complex 1. These electrons then participate in oxidation-reduction reactions inside complex 1, which produces the energy to pump 4 protons into the inner membrane space. Once completed, these electrons then flow to coenzyme Q. Finally, the NADH plus heads back to the TCA cycle to pick up more electrons. The next molecule to arrive is our new friend FADH2. FADH2 is oxidized and complex 2 is reduced. This releases the protons from FADH2 into the matrix, leaving FADH to run back for more electrons. Complex 2 then quickly passes these electrons to coenzyme Q. Coenzyme Q now transfers all these electrons to complex 3. More oxidation and reduction reactions occur within complex 3, and 4 more protons are pumped into the inner membrane space from the mitochondrial matrix. The cytochrome C protein now picks up all these electrons and moves them to complex 4. Complex 4 is the last stop for these electrons before they are handed off to water. Complex 4 passes the electrons through more oxidation reduction reactions and pumps 2 more protons into the inner membrane space. Once this is done, 2 protons and 1 oxygen atom are combined with 2 electrons to form water. This is where the oxygen from the air we breathe is used. 4 electrons with 4 protons and 1 elemental oxygen makes 2 water molecules. Oxygen is called the final electron acceptor. This is the end of the oxidation and reduction chain that these electrons have been riding since they were first removed from glucose back in glycolysis and the TCA cycle. Now that all the electrons have done their work and provided energy to pump protons from the mitochondrial matrix to the inner membrane space, we are left with a much higher concentration gradient. This gradient creates a proton motive force, which makes the protons want to head down their concentration gradient. Evolution has given cells a magnificent way to take advantage of this proton motive force: the ATP synthase. The ATP synthase is a massive protein complex with a quaternary structure that consists of 27 specialized proteins. These proteins form 2 regions, the FO and the F1 regions. The F1 region is hydrophilic, and it sits in the mitochondrial matrix. The FO region is hydrophobic, and it is lodged in the membrane at the border of the mitochondrial matrix and the inner membrane space. The FO region allows protons to pass through from the inner membrane space to the matrix. As these protons pass through, they turn the complex. Each turn causes structural and binding changes that lead to the production of ATP from ADP and inorganic phosphate every 3rd turn. So, using our count from a single glucose, we have gained 2 ATP, 2 GTP, 8 NADH, and 2 FADH2s. Since each NADH produces 10 protons, and FADH2 produces 6 protons, we get a total of 10 times 8 plus 6 times 2, which equals 92 protons for each glucose. Since 3 turns of the ATPase synthase yields 1 ATP, that is 92 divided by 3, which equals approximately 30 ATP plus the 4 from glycolysis. So we get around 34 ATP for every glucose. This number is a rough estimate, because biology is messy and not always 100% efficient. However, what this means is that the electron transport chain is fundamental to the production of energy and has allowed for all forms of multicellular life to evolve.